But right now, please join me in a warm welcome for Dr. Kildell.
Thank you. All right, well, welcome. And thank you for having me as part of your Saturday afternoon. I have two short stories to tell you as part of my presentation today. Hopefully each will answer this question, the first being what is synthetic biology, and hope you'll get a sense of what role this new technology might play in the future of the world we have and the world we see. And then the second, what is BioBuilder, which I hope will answer the question of what this technology can play in the world of education and the way we learn about science and engineering. So I'll start with what is synthetic biology. And the word synthetic really here refers to the idea that we are building things from biology by combining simpler parts together. It is a way of building with biology from the ground up. Not taking what we see and tweezing it apart, but rather taking what we know and how it works and piecing it back together. And so if we start to think about what makes life's simplest component parts, if we think about the world of chemistry, we know that there are elementary particles that when you combine them add up to atoms. Those atoms can be recombined into molecules, and those molecules turn into macromolecules, those DNA, the lipids, the proteins in our cells, so that each of these elements can be combined in novel ways to add up to new and important things that we rely on. So macromolecules are really these building blocks of life. And then to think about how we might be able to build with those macromolecules, we need to think about what we need to get them. And in a living cell, those components are really all encoded from a reasonably simple molecule called DNA. Simple in that it is made up of only 4 small building blocks, but in complex ways and in combinations that add up to all of the proteins, all of the macromolecules that we see. So the goal that synthetic biologists have can be to build living organisms using these simple components, the parts of DNA, where the hope might be that we could write a sequence of DNA and with that new sequence build a novel organism, build something that we have a reason to apply and think that it might do some good in the world. We are not really in the field yet at that reality. The idea that we could type out some DNA and build some novel organism is not where we are in this new field. What we usually do instead is that we modify the DNA that exists in living organisms already, and through that modification, we build new organisms. And so the synthetic biologists that I work with and that define themselves